those joining us, we are having a webinar with a simultaneous translation. So let's start. Good afternoon, everybody. My name is Anne Clémens. I'm the director of ZABR, which is the Rhône River Basin Workshop Area. I'd like to welcome you to this ZABR Coffee to discover ecology and evolution of groundwater. This coffee has been organized as part of the publication of the second Elsevier book that is dedicated to biodiversity in groundwater. So in a few words, a ZABR Coffee is a short video conference in order to discover a tool, a book, to share the results of a research project that has been developed by the scientific community, and to exchange in order to find out how this can contribute to watercourse management. So ZABR coffees are open to scientists, to the Rhône Basin managers and managers of watercourses, to design offices, and to partners of our network. This coffee is organized by ZABR-Graie. So ZABR is a multidisciplinary researcher network that works on the Rhône River catchment area. And Graie is an association whose aim is to accompany, to support the transfer of results produced by scientists to water stakeholders. This webinar is also supported by the RNC Water Agency, EDF, and CNR, which is the Rhône National Company. It fits within the Water Week, and it makes an echo to the World Water Day of March 22nd. As regards the program, the schedule, we will first listen to a presentation by the book editors, which is the opportunity of a nice conference in order to discover the richness and fragilities of groundwater and the role played by organisms living in this groundwater. So we're going to listen to Florian Malard, who is a fellow researcher at CNRS in Lyon. CNRS is the National Center for Scientific Research. We are also going to listen to Sylvie Rétaux, who works at the Paris University, Saclay, and Christian Griebler from Vienna University in Austria. Then we will listen to two testimonies, Pierre Marchet, who is the director of the Adour Garonne Water Agency, and Xavier Chevillot, the director of SEPANSO, a Federation of Environmental and Nature Protection Association. We have over 250 people who registered to this international webinar. So we've got people coming from all over the world, 13% of people worldwide, 87% of French people, and a quite balanced distribution between research centers, universities, local authorities, design offices and companies, associations, and state administrations, and also private companies. We would just like to remind you of a few recommendations for this ZABR coffee. So first of all, the webinar is being recorded with collected the agreement of all participants and speakers. It is then going to be broadcast on our YouTube channel. So together with Isabelle Costa, my Graie colleague, we will relay the questions at the end of the various presentations. So let's start. I'm now handing over to Florian Malard, who is a fellow researcher in Lyon at the National Center for Scientific Research. He works in the laboratory for river ecosystem. So Florian, you have the floor to start with your presentation. Okay, so let's go. I hope you're all doing well. So first question, why a book on ecology and evolution in groundwater? I think that this project probably started in a bar, in a coffee, and in the end, we are very happy to be able to introduce it to you today during this ZABR webinar. Since the first edition of the book in 1994, and currently it's the second edition in 2023, and it's really the opportunity to highlight the eco-evolutionary nature of groundwater And to us, it's very important because it seems to us that groundwater ecosystems may not receive the attention they deserve in terms of management and prevention. So the result of that project, and there you can see it, it's a collective work that's been achieved by 78 contributors that we would like to thank. These contributors come from 56 laboratories in 17 countries. The book entails 24 chapters that are divided into 3 parts. The first 3 parts, which I'm going to present myself, presents the characteristics, the general characteristics of these ecosystems, the processes driving the patterns of groundwater biodiversity and the roles of organisms. The second part is going to be presented by Sylvie and is dedicated to the principles of evolution in groundwater and the biological traits in groundwater. The last part will be presented by Christian Griebler and is dedicated to the effects of human beings on these ecosystems, to legislation, conservation, and the management put in place in order to mitigate such effects. The idea of this presentation is to highlight the key messages of the book. And the first one is that one. We're walking on a water-soaked sponge. Over 60% of continental waters are actually groundwater. The so-called modern groundwaters, so basically that are aged below 50 years, they represent only 2.2% of the underground fresh water. Now, if I take this 2.2% and I spread it evenly on the continent surface, I create a mass of water with an average thickness of 3 meters. So you can understand this map in various ways. The first one is to think it's good that this water is subterranean. It's groundwater, because if it was in the surface, we would all have to be equipped with gills. The second way to understand this map is this huge mass of water should regulate and interact strongly with the various components of the Earth. And here I'm talking about the hydrosphere, the atmosphere, the lithosphere, and the biosphere. And indeed, it is the case. As an example, the very first consequence when you pump in an aquifer more than reasonable, the first consequence is not so much that you lower the groundwater table, is that you increase the flow of water entering the aquifer and decreasing the flow that comes out of it. And therefore, the first consequence is to restrict the flow of the surface water courses. So therefore, groundwater is the most commonly freshwater habitat on Earth. And I quite like this sentence in the book that says, exploring the diversity of living forms in groundwater is to biodiversity science what observational astronomy is to space science. In other words, as we discover these ecosystems, we go from discovery to discovery. On Earth, there are rough estimations saying that we count 50 to 100,000 subterranean species, which is the case here, and it means that groundwater ecosystems highly contribute to the biodiversity in the freshwater on Earth, because today we know that there are about 125,000 species in surface freshwater. For example, in Europe, we know that the number of groundwater crustacean species is higher than the number of crustacean species in surface freshwaters. So we are living on a water-soaked sponge, and a sponge full of life. This life is organized in a food web. So at the basis, you've got prokaryotic organisms like bacteria and archaea who are going to use mineral and organic elements of the surface in order to generate biomass. This biomass itself is consumed by other microorganisms like viruses or protozoa, and all these microorganisms support a food web consisting of invertebrates and in some cases vertebrates. 80% of the biomass of prokaryotes on Earth is underground. So these organisms, some of these organisms are specialists in groundwater. So basically, these are those who necessarily carry out part of their life cycle in groundwater. And these organisms have remarkable traits that will be presented by Sylvie Rétaux afterwards. So here I'm talking about subterranean Galapagos in order to describe a remarkable characteristic of this biodiversity. Each aquifer can be considered as an island, which is colonized by surface organisms. And these populations follow their own evolution because these various islands are very shortly or not at all connected, hydrologically speaking. So a recent example, which is quite striking, is that of the calcrete aquifers located in the Western Australian desert, where each aquifer that you can see here develops in limestone deposits close to the ground. And each black island here represents an aquifer. There are dozens over hundreds of kilometers, and each island has its own set of species. This is a very concrete example, which is not exotic. I use it because it has enabled to highlight hundreds of species of beetles that are very little represented in underground waters. But in the same way, in France, the aquifers of the Jura, the Pyrenees, the Languedoc, or the Mercantour all have their own series of species and no common species between themselves. In order to understand the subterranean biodiversity, we need to look both at the past and the present. In Europe, the size of the distribution range of species is increasing towards the north, and it's very likely that the cold Pleistocene climates extirpated species with small ranges of distributions. And simultaneously, they also selected more mobile species that are able to recolonize deglaciated territories. On the other hand, looking at the specific richness of regions, this richness is at its maximum in mid-latitudes, precisely where the primary productivity in the surface is important. This primary productivity is actually a source of organic matter that penetrates groundwater in order to feed organisms living there. So organisms in groundwater have a twofold role. They act both as a purifier and as architects. This purifier role is very well known in the depollution of water table because the techniques used consist in stimulating bacteria in order to degrade pollutants like hydrocarbons. The architect role, or because we call them ecosystem engineers, it's because they keep restructuring the sediments in the aquifer, and basically they are the flow of water and solutes in the water. This architect role is somewhat difficult to understand for some of you, and therefore I would like to end this presentation with a short movie. So here you can see on this sediment columns that was reproduced in the laboratory, you have an amphipode that is about 8 millimeter, and you can see how easily it is able to move the sediments here, for example, with its front members in order to widen the space. And a few minutes later, as you can see, with its appendixes, the rear appendixes, it is able to move sediments again. So the researchers that have studied the influence of these sediments moving have shown that moving the sediments in this column enables to maintain the efficient porosity of the aquifer, basically the porosity that contributes to the flow of the water in the aquifer, in the table. Thank you very much for your attention, and I hand over to Sylvie now. Thank you very much. Thank you, Florent. We are now going to listen to Sylvie Rétaux, Research Director at the Paris University. She's going to continue with the presentation. Good afternoon, everybody. So the last slide shown by Florian is an excellent transition for me in order to tell you about the subterranean fauna and the phenotypic characteristics. So this subterranean fauna, whether vertebrates here on the top of the slide or invertebrates, as you can see at the bottom of the slide, this fauna have a troglomorphic nature in common. Basically, this means that they have lost all parts of their eyes and pigmentation. And in the subterranean environment and in groundwaters, it's a highly reproducible experience. All animals have what we call a convergence on these blind phenotype and depigmentation phenotype. And the book dedicates 4 chapters to animals, to subterranean animals, which have provided a lot of information on the evolution mechanisms in this environment. So like crustacean, the proteus as well, and fish as well, the CNRS mexicanus. So how do these animals lose their eyes? Basically, you have here a short movie about the embryonic development of a cave fish, Astyanax mexicanus. So this video was recorded in vivo, live, in order to show the development of the nerve systems and the head. So there you can see the nerve systems and the green cells on the side. This corresponds to the future eyes and retines of these embryon of cave fish. So it is indeed a cave animal that is going to be fully blind, basically with no eyes, but the development of eyes happens during the embryonic development, and it produces small and poorly shaped eyes. So here you have a picture after 1 day of development, and it clearly resembles a human pathology called coloboma, where the central part of the eye is missing, both in the fish and on the human. So this is just to illustrate that these animals who live in a subterranean environment are kind of natural mutants, and this can often be used as a biological model to treat human pathologies. So in order to understand how these animals lose their eyes, you can kind of rewind the embryonic development and compare it whenever possible between the development of cave animals and the development of animals of the same species or same gender who have eyes because they live in the surface in rivers, for example. This is possible here with the Astyanax mexicanus species, and it shows that there are many variations in the development of both fishes, both in terms of morphology, of molecule aspects, and transcriptomic aspects underneath. So this is a biological discipline called environmental evolution, and it enables to understand how the morphologic evolution may happen based on variations of processes during embryonic developments. And it also enables to illustrate the fact that there are techniques and powerful techniques and technologies in order to address such mechanisms. So in this case, it's a generation mechanism that happens at the beginning of the process. Basically, it's cell death very quickly after the embryonic development. So losing one's eyes is not that easy. I've just shown that these fishes start by developing eyes and then have these eyes degenerate. And this pattern is true for all species of vertebrates where we have performed studies. So here you've got 3 examples, the fish and the proteus, this cave amphibian. And this is just to show you that the eyes do develop in all embryons of these species, as you can see on the pictures here. So this means that the shaping of eyes is a necessary step during the development of the head. And we can easily understand that this is necessary thanks to the development processes. It also means that there are forms and anatomies that the embryonic development cannot produce, which is what we call developmental constraints at morphological evolution. And subterranean invertebrates are a very clear example of this idea of developmental constraints to morphological evolution. So basically, I was saying that these animals lose their eyes, but on the other hand, they win a lot, as I wrote here, because they lose the visual sensory capacity, but they develop sensory compensations. This is represented here with the gammarus and with another fish species. So on this crustacean, you have an increase of the size of antennas by 65%, which are the smell sensory receptors, sensors. And this is also illustrated here with the fish, with an increase in the number of neuromasts, which are also sensors capturing the pressure and vibrations of water. And there is also an increase of the surface of nostrils. So the nostrils are much bigger in the subterranean fish than in the river fish. So there are sensory compensation to the loss of vision, both at anatomic level, but also at behavior level. So subterranean animals are also fabulous examples to study what we call evolutionary genetics, because some of them have surface shapes and subterranean shapes, and they can be crossed. So this crossbreeding generates first and second generation hybrids. And thanks to these, we can implement quantitative genetics methods in order to understand which genes control the troglomorphic characteristics, like the loss of the eyes or the loss of pigmentation. And we can therefore develop genetic maps and study the transmission of these characteristics. Groundwater life is also enabled to work at the level of genomics. There are more and more genomes available for more and more of species. And here I wanted to show you a study that enabled to understand which genes can be lost or can become unfunctional. We call these pseudogenes after the colonization of the subterranean environment. So you can look at the two species circled in red here, the Lucifuga, which is a cave fish from Cuba, and the Astyanax mexicanus. And basically here, we've tried to look at the number of specific genes for vision. So genes that are used only for the development of the vision system and nowhere else in the organism. So we've compared the number of vision genes that have been broken in the genome of these two species. And for the Lucifuga, we identified 19 genes out of about 80 that were broken, although only one has been broken for the Astyanax. So this gives us precious clues on the time that these cave animals have spent in underground water, because the more time they've spent in underground water, the more genes have been broken and become pseudogenes. And then we can develop models based on that. So for Lucifuga, it was determined that Lucifuga colonized underground water about a few million years ago, although for Astyanax it was probably less than 20 years ago. So it just shows that it's very easy to kind of shape a cave animal. Underground waters are a kind of species factory. There have been many controversies in the past years on the way, on how the speciation happens in order to produce cave anthropomorphic species. So there are various hypotheses, some based on multiple colonizations and convergences that are repeated for various cave species, and this is opposed to the unique colonization and the underground speciation. So this is represented here in blue, showing the the new species after the colonization of the underground or of the groundwater. So basically, this shows that we have 3 different species of these beetles, underground beetles. So there are deletions in the genome that are exactly identical to all 3 species of Parosta. And this shows that the second hypothesis, that is the correct one, so the unique colonization, because the probability to obtain exactly the same deletions in the same places within 2 different species is almost null. So this shows the identical deletions with various types of beetles. So as a conclusion, I wanted to propose various evolutionary forces in action, or evolution mechanism in action in underground water. First of all, the so-called neutral evolution with genetic drift. Basically, it means that in this case, having eyes or not in the underground environment gives the same likelihood of survival, and the selection relaxation enables an accumulation of mutations. This is what we call neutral populations by genetic drift. There is also an evolution by selection, by a selection mechanism that is potentially a direct evolution with selection against the eyes. So basically, selection against the eyes means that in the underground environment, having no eyes could be an advantage, and there would be an accumulation of mutations and a loss of eyes by a counter-selection processes in the detriment of eyes. This experience has been has shown that maintaining eyes in the subterranean environment is very costly in terms of energy. So yes, and there's a third possibility, which is evolution by indirect selection by pleiotropy, which means that it's not the eyes which are selected, but the increase in sensory mechanisms or sensory compensations, like the olfactive system, for example. So in this case, this would mean indirect selection, and that eyes were lost as a kind of side effect of the increase of another sensory mechanism. And this happened through a pleiotropy process, because during the embryonic development, the same genes are used to develop both systems. The very last conclusion sentence was a quote by François Jacob, who is a Medicine Nobel Prize winner, who basically said that biological evolution is a kind of evolutionary DIY, and by reusing the same molecular mechanisms, this evolutionary DIY is a major process in the underground environment. So that's all I wanted to tell you. Thank you very much, Sylvie. We clearly understood the message. So thank you very much. So we now hand over to Christian Griebler. He's a researcher at the Vienna University in Austria. He's the 3rd editor of this book. So Christian, you have the floor.
So welcome to the audience. It is my responsibility now to, or my pleasure also, to introduce you to the 3rd part of the book, which is a more applied one. Although these ecosystems are below our feet and quite hidden, they are nevertheless exposed to threats. We need to manage them, we need to protect them, and for their protection we need a legislation, of course, which is appropriate. And this I will talk about in the next couple of minutes. We all need groundwater in the one or the other way. So either we drink it because groundwater is used for drinking water production, or we use it for irrigation in agriculture, as solvent in industry. So there's so many uses of groundwater, but we always forget this is also an ecosystem where animals live in, and Florian introduced this to us quite well. Groundwater ecosystems, like aquatic ecosystems at the surface, really face critical times, so to say. We are living in the Anthropocene. That means we make use of all the resources of our planet more than there is a renewal. So we make use of it in an unsustainable way. And the same is true for groundwater, of course. There is overexploitation, so we deplete our reservoirs. Climate change has a dramatic effect also to our groundwater resources and to groundwater recharge, the renewal of groundwater. We face a warming also in the subterranean realm. It's not only happening at the surface. There is ongoing pollution. There was pollution already in the past, but pollution changed, and there is salinization. So groundwater, specifically near the coast, become more and more saline because we overexplore our freshwater reservoirs. Related to the pollutants, and this is maybe of interest, when you look into the media, you hardly read about contamination of groundwater by oil products. This was 20, 30 years ago, but nowadays we read about microplastics. We read about specific pesticides or pharmaceuticals that are found in groundwater, antibiotics, for example. Or recently in the media, you find compounds like the per- and polyfluoroalkyl substances that are used manifold in industry that are found in groundwater. Yeah, the quality and the quantity of groundwater is endangered, and changes in quantity and quality, without any doubt, of course, affects not just the resource, but also the living space, the ecosystem, and the biodiversity down there. The book provides a lot of information related to that. For example, a chapter that deals with the sensitivity and tolerance of groundwater organisms related to many different pressures and contaminations. So what I mean is chemicals on the one side, but also warming on the other side. And here in this chart, you see an example that surface organisms like an amphibod that is shown here has a much bigger tolerance to a temperature change in water and is adapted to a larger breadth, so to say, in temperature, while many groundwater organisms are very specifically adapted to their environmental conditions. And for example, in Central Europe, this means a quite stable and low groundwater temperature. And if we now change this temperature, either by introducing warmed water or also the changes going on by climate change, this is, of course, endangering this groundwater fauna. We also found out in the past, and that there's not many data available, we also need to stress that in some aspects, our research is still producing highly needed, urgently needed data, not yet available, that groundwater organisms are not all the same. So they have very different tolerances to stressors like contaminants. Here, an example is given with zinc as a heavy metal. Yeah, so we need to protect these underground ecosystems the same way we do with the surface aquatic ecosystems. And I think we all agree, we even are not doing a very great job in protecting surface ecosystems, but we hardly protect subterranean ecosystems so far. I listed here a number of directives and conventions in place that have been developed to either protect areas, all ecosystems, or the biodiversity or individual species. And if we have a look into these conventions and we search for the protection, the specific protection of groundwater ecosystems or groundwater species living in groundwater, then we hardly find anything. This is the main message of this slide, so you don't need to read all the details. Within the individual conventions, there is only in exceptional cases groundwater fauna and biodiversity is considered. In Europe, we have the EU Groundwater Directive that was set in place to protect and guarantee a good quality of groundwater. And also here, it's not necessary that you read any details. What I want to tell you is in the first 2 pages in the introductory part of this EU Groundwater Directive, which was released in 2006, 2007, there's a lot of information given to the chemical status of the groundwater, which should be good, the quantitative status of the groundwater. So there should be enough groundwater. This is things we need to take care of, but there's hardly any information about the organisms living there and the status of groundwater as an ecosystem. There's one small section where it is stated that research should be conducted in order to provide better criteria for ensuring groundwater ecosystems quality. And where necessary, the finding shall be implemented in the revision of this directive, which is now the case at the moment. We need to revise this directive. And it is also stated, which is quite interesting, that such research needs to be encouraged and funded. So the European Union, as well as the individual nations, have a responsibility to put money in that. And we have in the audience today persons from stakeholders, representatives from authorities. So just to tell you, groundwater ecologists haven't been lazy in the past 10 to 20 years. There have been a lot of tools developed, and this is just some examples provided here. So sampling methods have been standardized. There was early development of classification schemes for groundwater ecosystems. People looked for appropriate ecological criteria and bioindicators to be used to assess the status of groundwater. And of course, also to delineate the reference conditions, which is always something needed when you do an assessment, and to develop some indices. So the book with 2 to 3 chapters provide information on how a classification of groundwater ecosystems at different spatial levels can be done, from the local, regional to the continental level, and how to proceed. It also gives examples of classification systems that have been developed in the past. You see here the groundwater habitats classification done by Canu and colleagues from France, developed 2013. And for individual parts within Europe, other types of classification systems, some mainly related to the structure of groundwater systems, others mainly related to the composition of groundwater fauna and biodiversity. So these systems have been developed, and of course they need further development, but it is always necessary to tell people from the applied side, from water economics, that there is already tools in place that can be used already today. Yeah, when assessing groundwater ecosystems, it is important that we do not look only into the structure of these systems. So do we have porous or karstic or fissured aquifers? It is important that we do not only look for the quantity and the chemical composition, but include ecologically sound parameters. And a few criteria are listed here and provided. So it is important to look who is living there. It is important, is there many organisms Do they have a high or a low activity? Is this typical for this kind of ecosystem? How are these communities organized? Do we find organisms in these communities that do not belong there? All these questions need to be addressed. And there is assessment schemes available, and the book provides sufficient information on that. Yeah, the tools we have in hand, we may use groundwater fauna, invertebrates seen here, top left, as bioindicators. We might make use of microorganisms in assessing groundwater quality. There is already equipment available that can be used for ecotoxicological investigations. And of course, we might take a look into the groundwater chemistry, but also here looking at parameters that are of ecological significance, like nutrients or dissolved organic carbon, dissolved organic matter. That is the direct energy for the microbes in groundwater. On the right side, you see an assessment scheme, how it has been developed in different ways, in various ways in the past, but an example is provided in the book, how you can start assessing your own groundwater, being a waterwork, for example. So with easy parameters or easy to measure parameters, with looking for reference values in the literature, coming to an early conclusion. And the moment you find that something is going wrong, you go into a second tier where you follow a more expertise intensive evaluation of your groundwater ecological health, so to say. There is a number of indices that have been developed and evaluated in the past, like a fauna index, but also microbiological, ecological indexes can be found. Maybe the final aspect to be mentioned is we need to take care of our groundwater biodiversity and these valuable ecosystems in the near future more intensively in terms of legislation. But before we do that, we also need to identify areas and sites and places that really needs to be protected first. And this is not so easy because you can follow different strategies for protection related to aspects of groundwater chemical quality or groundwater ecological quality or ecological health. The book provides with a chapter dedicated to that, a review, a summary of strategies you can follow. You can either follow the way that you want to protect the highest number of rare species available in systems. And you see here, one can look for the species numbers and look at individual species you find at different sites. And then you choose for a combination of sites that allows you to protect most of the species you have available. But your strategy can be completely different. You can go for high abundance of organisms. You could go for endemic species. So this is then up to the researcher to choose and further develop these protection strategies. And there is ongoing discussion, what is the right way. One way I really also support highly is that we do not look only to subterranean ecosystem, but we look at them always in the combination to surface aquatic ecosystem and also surface terrestrial ecosystem. So in this map, you see several ecosystems connected so far, and we can then produce layers, surface layers and subterranean layers, and we can then start to combine within these layers either the river systems together with the wetlands, the river system with wetlands, and then the aquifers below them, and look for the places where we give them a prioritization in protection to choose for the best and the ones we need to protect and conserve fast. Because you all know there's never enough money and there's never enough area available to protect all land. Of course, it would be a vision, of course, but that's not the case. Yeah, the book, as I said, provides many chapters also related to applied aspects. So I invite also people from authorities, from the applied sector, water sector, to, of course, buy the book and read these chapters. And together with the applied chapters, also read all the other chapters dealing with basic science. With this, I thank you all for listening.
Merci beaucoup.
Once again, we need to thank all the authors and co-authors, the publisher. And yeah, no book can ever be finished. This is a phrase of Sir Karl Popper that says, while you work on the book, things are changing already. And the moment you are done, you would need to start again. And with this, I thank you for your attention.
Thank you very much, Christian, for your presentation. I'd like to invite you to stop the screen sharing, and in order to start the discussion, I invite you to switch your camera on if you want to, and I would like to give the floor to Pierre Marchet, who's director at the water agency Adour Garonne, and who has promoted the implementation of indicators in order to assess the groundwater quality. Can you tell us about your experience, Pierre? Yes. Can you hear me well? Okay, I would first like to mention that water agencies are not responsible for the study of groundwaters, but their mission is to protect them. And in order to protect something, it's difficult to protect something that you don't know. Although for other aquatic ecosystems, we have a lot more knowledge, much wider knowledge. So we started to improve our knowledge. And in the 90s, we could see diffuse pollution arriving in the water table, and especially pollution with nitrates with very high concentrations. And in the Pyrenees, there was a laboratory studying underground ecology that started studying the caustic systems. And the idea was to try to look at other types of aquifers, not too far from the laboratory. There was this alluvial water table that was highly impacted by nitrates. So we started performing a thesis, well, writing a thesis. It was funded by the laboratory, and we wanted to see how these organisms, subterranean organisms, had been impacted by this high nitrate pollution. And we could see that there was not so much of a sensitivity to this pollution. But in the same area, once we had managed to identify the species, we decided to find whether they were impacted by mineral micropollutants, but it was still not so good in terms of indicators to show the relationship between the pollution and the species. A few years after that, we had the opportunity to conduct a study in the same area, and we studied the impact of organic micropollutants and phytosanitary products in association with the Lyon laboratory. And there we started having interesting results showing a relationship between the pollution by phytosanitary products and subterranean animals. Simultaneously, we tried to promote these with various stakeholders. It's not easily done to have people understand the existence of subterranean species in aquifers. Even the questions here in the chat show that this is still not very well known. For us, there are also interests and advantages, and we were interested in the relationship to this water course. It was difficult for us to identify good indicators, and we noticed that things were happening in the Rhône River basin, and in our ZABR, so in our water management, water development and management master plan, we are trying to promote the regulation of the annual water cycle. And in our ZABR, so in our master plan, it says that we have to look at the impact of these things on the styger fauna. So I think it's a new progress, and we could observe that part of our problem contrary to the Iberian rivers, is to determine the species. So it's still a matter of specialists, and it's going to be a major obstacle for quite some time again. So we are trying to promote this at national level, because this is not so much the role of water agencies to perform research on topics that go beyond their territories. But right now it's difficult to have our voice heard, and the directive mention clearly says that we should do things, but there's not so much that is currently done on the field. But we want to remain positive and hopeful. Thank you very much, Pierre. So in terms of Steigerfauna, we have also Xavier Chevillot here, who's the director of SEPANSO, who is performing this Steigerfauna program in the New Aquitaine region. So what can you tell us, Xavier, in this respect and as part of your program? Yes, good afternoon, everybody. So my name is Xavier Chevillot. I'm the director of SEPANSO, which is the Federation of Nature Protection Association. Very quickly, before I introduce the SEPANSO, I would like to thank the three speakers, as well as Pierre Marchet. It's so interesting. Thank you so much for your contribution and for your high quality presentation. It's very comfortable to have the opportunity to interact with such experts. So why am I here today, and why does SEPANSO pay attention to the Steigerfauna, and how does it echo today's presentations? Well, as a Nature Protection Association, SEPANSO has three missions, or three key objectives. The first one is to be a watch body for nature, a watch body for nuisance on the environment and in the various environments, both surface water and groundwater. So we are indeed taking various warnings of pollution into account in our activities. Our second mission is to protect environments, to manage environments, and to try and conserve, protect the biodiversity. For 50 years, SEPANSO has tried to develop natural reserves. So we are managing 3 national natural reserves. So obviously this echoes the protection of environments and the protection of underground environments, because indeed, and we know that there is still a lot to be done in this respect. Now, the third mission of SEPANSO is to share projects and to coordinate projects and programs, knowledge programs in the field of biodiversity, because the better you know, the better you protect. This is really the red thread of this presentation. So therefore, it's highly legitimate. And by talking about a specific program, which is the Steigerfauna program, which has been coordinated by SEPANSO since 2022. It's a very ambitious program, which has 2 objectives: that of knowledge, the taxonomic knowledge, basically this is about better knowing subterranean communities, and there is also a bioindication program. So what could be the relationship between the current biodiversity and the quality of groundwater? This is really what's driving us in this program. It's a highly ambitious program. It's probably the most ambitious one today in terms of surface of territory covered, because it covers the entire New Aquitaine region, which is a vast territory. It's almost a quarter of the French territory. And therefore, it's a very ambitious program that is going to open almost 300 sampling sites in this area. Very quickly, I would also like to thank Pierre Marchet and Florian Malard, because I work in close collaboration with Florian as part of this program, and also the funders, because some of them are here today. So not only the funders, but basically all the stakeholders, like the Adour Garonne Water Agency, who contributes to the funding of this program and has provided their support from the very beginning. as well as the Nouvelle-Aquitaine region, the Gironde department, the FEDER European Fund, and a private sponsor as well, LEHNA. So I would like to thank all the stakeholders, and that was it very quickly for my testimony today. Earlier, we're talking about expertise in the field of biodiversity in order to move towards bioindication. And today we have parts of the skills in our team, but it's not enough. And we are always looking for wider skills, wider competences at European scale. And I will stop here. Thank you very much, Xavier. I would now like to have 15 minutes for a Q&A session. So short questions and short answers. In the chat, we have questions regarding the localization of these species. Can you find these species in all groundwaters or only in the caustic groundwaters? There is another question in this respect. Can we find all these organisms in all types of aquifers? Is there any speciation between the various groundwaters? Anyone would like to answer that question? Florian? So in the Rhone, groundwater, there are numerous species. There is life in groundwaters anywhere where there is space to live. As soon as there is water, there is life. To give you an example, nematodes were discovered in 2011 at a very deep depth in mines in South Africa. Now, very concretely, the deeper you go, then the diversity decreases.
Vont s'allonger et plus vous avez la possibilité de tomber sur ce qu'on appelle des eaux
because there is less organic matter and there is a higher likelihood to have anoxic waters. So it's true that the high diversity of groundwater is mostly located in the upper parts of aquifers, whether they are caustic aquifers or not. Are there clearly identified model and sentinel species in groundwaters? Model species, yes. Basically, this is what we call, this is the species we use in a repeated manner to understand a certain number of things. We talked about the Astyanax, the Azonis aquaticus. Sentinel species, it's a little bit more difficult. Why? Every time you move from one aquifer to the other, the assembly of species changes. So it's difficult to have one sentinel species for the entire Europe or French territory. I'm sorry, we mostly use surface species that can be introduced in pits in order to measure the answer of such organisms. So in the book, are there illustrations of subterranean species in volcanic aquifers, for example? Is there a worldwide vision? I'm sorry, there's no specific focus on volcanic fauna. It's true that depending on the type of lava, there are subterranean species that will develop of many kinds, arthropods, millipedes, and so on and so forth. But there is no chapter specifically dedicated to this fauna in the book. Sorry about that. Christian was saying that a book is never finished, so maybe it's a new chapter to write. How important is the porosity of subterranean soils? What is the analogy between fresh water in the surface and wetlands? So the first influence of porosity on organisms is basically, there are two things. You've got the pore size that dictates the size of organisms that live in this environment. So you can have fishes in karstic areas, but it's going to be more difficult in alluvial areas. Without porosity, which is important, what is important for organism is the porosity that is connected. So basically pores that are connected and that let the water flow. And when the water flows, organisms can circulate as well. So the second idea that is important is this efficient porosity idea. And third, it's more a matter of permeability. So basically, how does the water circulate between the pores, and how is it connected to the surface water? So when it's well connected, they receive a higher quantity of organic matter. Okay, and underground, well, groundwater ecosystems, are they more resilient than surface ecosystems? Maybe, Christian, you would like to answer that question?
Yeah, sure. The idea is that every ecosystem, like we human beings, when we are strong, when we have a lot of energy, we can withstand disturbances and threats. And groundwater ecosystems very often are depleted in energy because the energy stays at the surface, and with depth, the energy becomes less and less. So one sign of it is that you have less organisms that are less big, and they are not so frequent. So a pressure, a disturbance, contamination of these systems lasts very long, and the system itself cannot withstand it evenly than surface systems. So they are less resilient, so to say, also less resistant to the disturbance, and disturbances last very long in the subsurface.
Are there ecotoxicological tests in order to determine the disruption of these groundwater?
Yeah, as I tried to introduce, we lack very often groundwater specific information. That means ecotoxicological tests that have been conducted using organisms that live in groundwater. Very often, a contamination scenario found in the groundwater is tested in the lab using surface water species like the Daphnia test or fish egg tests or fish larvae tests or whatever. So the information we have is not very precise, I would say, because groundwater organisms live in a different environment, are getting much older, sometimes 10 times older than their surface water relatives, and they tend to accumulate chemicals in their body. They are of a lower activity, physiological lower activity and metabolism than their surface water counterparts. So it is really hard to judge on these tests we have available how these organisms experience the disturbance. So we are still in need of more data. There is a few data existing that are summarized in the book, but in the future, we will need to develop specific groundwater ecotoxicology tests and procedures to provide the right data.
Another way to look at the biodiversity of species in groundwater is to find out the services provided by these organisms, for example, depolluting groundwater. So for what kind of pollutants are they efficient?
Maybe I also take over this question. Yeah, microbes are fantastic, genius organisms, and they can deal with almost all kinds of contaminants. However, human beings started to develop compounds, chemical compounds that are not biologically cannot degrade it biologically anymore in a reasonable time frame. And also the microbes in the groundwater cannot help here. In the recent past, we saw that groundwater microbes and microbes all over the world can help if there is a contamination by oil or organic solvents. But the compounds we find nowadays, like microplastics, or the PFAS I mentioned, these pair and polyfluorated compounds, or some pesticides that are designed not to be degraded fast. They are there in very low concentrations, and this kind of contaminations, the organisms in groundwater can hardly help and provide this service of water purification, unfortunately. But the same organisms or other organisms at the surface also can't help with this kind of chemicals.
Christian, another question in the chat. could we make a difference between various subterranean sites for preservation purposes? Do you mean exploitation sites? I think so. I'm not so sure that I get the question right. Well, as far as I know today, I'm talking about pumping sites. I don't know any single example in France of pumping sites that could have been modified because of the presence or of the existence of subterranean fauna. What's important to understand, though, when a fauna is existing in groundwaters, it is more present in the groundwater, but not only close to the pumping site. So we're not going to move a pumping site just because there is one specific species in the groundwater, but it could be considered we could consider adjusting, maybe adapting the pumping modalities in order to try and mitigate the impacts on the ecosystem. I guess we're going to stop here. I would like to thank you all for your contributions and presentation. We have a few key messages that we can take away. Basically, we must take the biodiversity of groundwaters into account. It takes specialists to determine all these species, so we need people who are trained for this. And we need to protect and preserve this groundwater biodiversity because it provides services to us human beings. I would like to thank Florian, Christian, Sylvie, and our two, so Pierre and Xavier, who provided their testimonies. Thank you all for your respective contributions and presentations. So thank you very much to all of you. I wish you a nice rest of your day. And we would like to thank the interpreter as well. Thank you very much. Bye-bye.